Hi everyone, thank you for coming today. My name's Ethan Ribblesworth.
I'm Molly Meadows.
We are both graduate students from the University of Exeter in the UK, and we're here studying our Masters by Research, looking at stingrays and dietary analysis. Through this, we've also been co-advising with Dr. Owen O'Shea on the fall 2016 Stingray research team, and we're incredibly proud of what they got to present for you guys today. So without further ado, let's pass it on to them, give them a warm welcome.
Good morning, everybody. Thank you all for coming. My name is Ellie Jones.
I'm Keenan Gamble.
I'm Garrett Pilgrim.
I'm Claire Tan.
I'm Neil Connaughton.
I'm Ethan Skelly. I'm Jake Weinstein.
And we're the Fall 2016 Stingray Research Team. Today we will be looking at a dietary assessment of two sympatric species of ray, or two species that occupy the same niche. Rays evolved over 450 million years ago from the late Silurian era, making them older than trees, and since then have radiated to occupy every major aquatic bioregion on Earth. Rays are also critical to the overall health and function of these ecosystems. Rays represent a significant portion of fish biomass in tropical coastal environments and typically characterize marine nearshore habitats. By their very nature, coastal environments face continued extrinsic pressures on account of being the interface between terrestrial and marine habitats. This includes habitat degradation as well as human-induced disturbances such as overfishing, pollution, and urbanization. Because of this, it has never been more critical or relevant to establish a baseline for the conservation and management of these vulnerable habitats.
In the coastal habitats of South Eleuthera, we are studying two species of ray. The southern stingray, Dietary americana on your left, and the Caribbean whiptail, Styracosaurus smarty on your right. In Eleuthera, these two large-bodied species coexist in the same environment, and we are trying to see if they partition or share resources to continue to coexist. Understanding the sharing and partitioning of resources is the first step in the conservation process because we first have to understand these animals in order to know how to protect them. Our objectives. First, we are addressing the issue of data deficiency in this believed to be keystone species. Our overall objective is to determine the dietary content and preference of these two stingrays and establish which ecological gradient, space, time, or resources is being used to combat competitive exclusion, which is when one animal is driven out of their habitat or to extinction. More specifically, our objectives are to compare the diets of these two stingrays so we can better understand the habitat in which they live.
Materials and methods. Here you see a map of Eleuthera. There, this star right here is where you're sitting today at the island school. For southern stingrays, we travel to the Schooner Keys and the surrounding sandbars, which are located here, to capture these rays. For Caribbean whiptails, we travel to local creek systems, such as Plum Creek and Deep Creek, and they're also found in the northern and southern exumas. To capture these rays, we form one line of people off the bow and one line off the stern of the boat. Once in the water, we encircle the ray, and then we constrict the circle to herd the ray towards a 10-meter seine net. Once the ray is in the seine net, we then use a dip net to fully constrain the ray. After this process is done, we restrain the barb of the ray for both our safety and for the ray's safety. After this is done, we take morphometric measurements and perform gastric lavage.
Gastric lavage is the invasive yet non-lethal method we use to assess the stomach contents of rays. This process involves the flushing of a ray's stomach contents to collect data. First, the ray is inverted with its pectoral fins pinned to induce tonic immobility or a state of natural paralysis. Next, a 60 milliliter syringe fitted with a long silicone tube is measured against the ventral exterior portion of the ray, as you can see on the screen, from the buccal cavity down to the stomach gut. The tube is then inserted through the buccal cavity, down the esophagus, and into the stomach gut region until resistance is encountered. Next, 3 60 milliliter portions of salt water are injected through the tube and into the stomach to induce lavage, while the anterior portion of the ray is lifted onto a porous container fitted with a 1 millimeter mesh net. All biomaterial collected on the net is then transferred into transparent sample vials, which are placed on ice to retard digestion on our way back to the lab. Additionally, we do collect samples that are produced by defecation due to unnatural stress placed on the ray.
Once we are finished in the field, we return back to the lab and sort through our stomach contents using a 3-layered sip stack. While we are doing this, we also rinse our stomach contents out with fresh water. Then we put each layer under a dissection microscope in order to identify the different dietary components to the highest taxonomic level. Then we are able to calculate the prey accumulation curves. And this shows us that the sample size collected shows an accurate representation of the diet of the species of stingray in which we are studying. Then we also calculate the percent IRI, or percent index of relative importance. And this encompasses all the different calculations from which we collect during our data process and puts them up into this formula shown up on the board. And this formula encompasses percent frequency of occurrence multiplied by percent numerical abundance plus percent mass. And the percent index of relative importance tells us the most important prey types in this species of stingray. Our results. So in total, we captured 31 rays, 21 of them being southern stingrays, and 10 of them being Caribbean whiptails. And 60% of the southern stingrays assessed had stomach contents, and 70% of the Caribbean whiptails assessed had stomach contents. And in our study, we aim to measure a range of disc width in order to show an array of life history stages within these rays. And the range disc width, or the maximum range of the animal, is shown on this ray, is from here to here. And our range disc width for southern stingray was 460, 952 millimeters, and our range disc width for the Caribbean whiptail was 434 to 628 millimeters. Once we are finished collecting our data, we put them up into these 3 graphs shown up on the screen. And these graphs are organized through rank abundance, and on the x-axis we have the different prey types, on the y-axis we have the total abundance. This graph encompasses all the different stomach contents assessed, both within the Styracosaurus smartae and the Dietary americana. And what is really interesting to look at is that the most abundant prey type is Alpheus, which is a type of snapping shrimp, and there's a photo of it shown up on the board. However, when you look specifically at the different species of stingray, you will see that in the Styracosaurus smartae's diet, Alpheus is ranked 2nd in abundance, and in the diet of the Dietary americana, it's ranked 4th. And what this tells us is that the Alpheus makes up a big portion of the diet of the Styracosaurus smartae, but not as big of a portion of the diet of the Dietary americana. Another really interesting thing to look at is that 6 out of the 12 prey types are represented in the diet of the Styracosaurus smartae. And what this tells us is that the Dietary americana has twice the dietary diversity as the Styracosaurus smartae.
This table shows the index of relevant importance for the 3 major prey types found in the stomachs of the Styracosaurus smartae and Dietary americana. There are teleosts, which are bony fish, crustaceans, which are crabs, shrimps, and other prawns, and annelids, which are worms. One interesting statistic is that crustaceans make up above 90% IRI for both species of stingray. And another interesting statistic is the complete lack of teleosts in Styracosaurus smartae's diet. Discussion. By analyzing the index of relative importance, we have found that crustaceans are likely to be the main prey type for both species of stingray. There are slight differences in the diet between the 2 species, such as an elevated presence of annelids and no teleosts in Styracosaurus smartae's diet, and fewer annelids and more teleosts in Dietary americana's diet. This is similar to observations noted in past studies conducted in Ningaloo Reef, Australia. This is a foraging strategy plot, which is designed to see the foraging mechanisms that stingrays employ when feeding. We graphed each of the 3 major prey types for both species, and on the x-axis is the percentage frequency of occurrence expressed as a decimal, and on the y-axis is the percentage prey-specific abundance. By comparing it to the Costello method here, which was developed for a past study, we can see that stingrays use dominant and specialized feeding strategies on crustaceans, and more generalized and rare feeding strategies on teleosts and annelids. But in terms of dominance, the positions of teleosts and annelids are swaps between the 2 species.
It's important to note that our study is the first comprehensive assessment of diet
ever conducted of assessing these two stingrays. And our research has never been more relevant, as during the course of our research, one of the species we were focusing on was reclassified taxonomically from the Himmenschurschmarde to the Styracuroschmarde. This illustrates how pressing our research is today. More specifically, in our study, we found that crustaceans dominate over 90% of the diets of both species, and because of this striking similarity in the diet, it is unlikely that dietary partitioning is occurring between these two stingrays. It is, however, likely that small-scale spatial partitioning is occurring, and we deduce this from the fact that the diets are so similar and the close proximity we have found these rays to each other. In the future, we can address possible biases in our study. For example, our sample size was not large enough to produce an asymptote on the prey accumulation curves mentioned by Neela. What this means is that we need to increase our sample size in order to accurately reflect the entire species diet. Also, we can conduct an assessment of prey abundance in the location of capture for our stingrays, and we can compare this data with the data of the stomach content in order to possibly explain the crustacean-dominated diets. Finally, another bias in our study is that because crustaceans have a hard-bodied exoskeleton, they resist digestion in the stomachs of the stingrays longer than soft-bodied organisms such as annelids and teleosts. What this means is that there's a larger window of time for us to capture and assess the rays and find these hard-bodied exoskeletons, and this can skew our data slightly. We plan to combat this by using other dietary analysis strategies, such as stable isotope analysis, and this will help us to remove the biases entailed with the use of gastric lavage. We would like to acknowledge the interns and institutions that supported us. This is our literature cited. Thank you for your time. Do you have any questions? So I believe the question was possibly why were there a lack of teleosts in the diet of the Styracuroschmarde? That's an excellent question. Actually, the lack of teleosts or bony fish in the diets of the Styracuroschmarde, it's because the Styracuroschmarde that we captured and assessed were found in local creeks, as said by Garrett, and there's a lower presence of bony fish in these creeks. And actually, the rays, we believe they don't specifically target bony fish. However, the fish will hover around the ray during feeding processes for a possible secondary predation opportunity, because food scraps are being tossed up into the air during stingray feeding, and it's possible that these fish get ingested accidentally. But because there's a lower presence of them in the creeks, that could possibly explain why we didn't find any in the diets. Yes?
So are the creeks and the dunes considered part of the same ecosystem? And did you see any crossover? Did you see any southern rays in the creeks or whiptails up in the dunes?
That's a really good question. So the question was, do we see any crossover between southern stingrays in creeks or Caribbean whiptails out at the Schooner Keys? And yes, we do sometimes see crossover between the two, but mostly they're in these areas. And the difference between the two habitats is we consider the creek system's inshore habitats, and we consider the schooner keys and the surrounding sandbars offshore.
The creek could also be like a nursery for rays. That's why we didn't capture as many large stingrays for the Styracosaurus smartae in the creeks, because they could be a possible nursery. Yes?
I was wondering how successful you were in capturing stingrays that you found.
So the question was, I believe, like, of the stingrays we saw, how many did we capture? I would say that about like 4 out of 5 stingrays that we saw you would get is about I would say. Yes?
So what was your, you get the stomach contents of these stingrays and you have to identify them. So what's the process of identifying them? I mean, especially if it's just the stomach contents of the stingray, trying to do it by species.
So the question was, what was the process in identifying different prey types? And so what we do is we start off is we take them all from these transparent sample vials and we put them under these sieve stacks, and what this does is kind of rinses out all the excess and all the excess material that we collected during in the field. And then we take each layer and we put them under this microscope and we look at them and get the various characteristics of them. Then we discuss them within our team and see what we say, like say if it looks more like a shrimp, we discuss and see if it's a shrimp or a crustacean. And we talk about it, and then we also get books and identify what we think the closest like prey type we think it is, and then we put it into our data. So that is the process. Yes?
You said you secured the bar. Talk about that. How did you keep it?
So the question was, how do we secure the barb? So we take a wet t-shirt and then we wrap the barb with the wet t-shirt and then secure that with Velcro straps.
Yes, do you have a question?
Do you guys have any insight to the range of their habitat in scale, miles, or meters? Do you guys have any?
The question was, do we have any idea of the range of the habitat, perhaps mileage or a scale? And actually, that's a really interesting question because this elasmobranchs as a whole, and more specifically stingrays as well, are classified as data deficient, as Keenan mentioned. And what this means is we don't know the basic life history, characteristics, range, and we don't know enough about the population of the rays to actually classify them as endangered or a stable population. We don't know where they are completely. We know that they are in the Bahamas and they have been seen in Central America, but we really don't know, and that's why this research is so important.
Oh yes, we do tag the rays upon capture. We use a pin dart to tag one of the pectoral fins of the ray. This is so we don't have any pseudo-replication of results, which means like if you see a tag number and then you capture the same ray, say the same day, you don't want similar results, which will skew the data.
And actually the two types of tags that we normally use on the rays are a pit and a dart tag. And we found that we don't actually use pit tags anymore on Styracosaurus smartae because their skin was too thick to get a reading through.